Metabolism and nutrition can seem daunting at times for a lot of med students, nurse students, all the students, in fact, all science students, because it's a lot going on. There's a lot of small details, and it's all molecular or micro in nature. Fortunately, we are going to go ahead and just touch on the very basics. That way, you have a nice layout. When you do arrive to nutrition, you'll be a-okay. When you get to pathophysiology, you'll be okay as well. And pharmacology, too. Pretty much everything lies in the metabolism and nutrition portion, or at least has some type of hand in the nutrition portion. So we're going to be talking about just how the body handles things. What does it break down? Why does it do it? Why does it need it? All that stuff. And easiest way to go is just to first define some really basic terminology. Anabolism. Anabolism means to create and make. When I think of anabolism, I think of my dear old Aunt Anna. Always so willing and loving and caring, and trying to take care of me and my siblings and knitting those pretty little scarves and sweaters and all that stuff. And all her love and care, dedicating so much time to build things up so we can use them. Such a good old Anna. Aunt Anna. While usually those things that interact or mean some type of buildup that you'll see when we see words or when we're talking words in metabolism, you're going to see genesis. Genesis means to create, while neo means new. On the other hand, on the flip side, Aunt Anna didn't really have the nicest of cats. Cats break things down. Mean old cats trying to break things down, shredding and poor old Aunt Anna's sweaters apart and blankets apart and all that stuff. Mean old kitty cat. If you were to see a word that means or has some type of role of catabolism and it has lysis at the end of it, then you're going to just please jump to the conclusion that it is referring to the breakdown of something. Lysis means to break down. So hemolysis means to break down blood. Cellular lysis or anything lytic in that nature just means to break down. Now, why break things down? Really, it's to make energy, and energy is usually stored up in these three forms: FADH2, NADH, but primarily this one right here, adenosine triphosphate. NADH means nicotine adenine dinucleotide hydride, while FADH2 means flavin adenine dinucleotide dihydride. Lots and lots of fancy words. But both of these will, for us, in this instance, if you decide to go into deeper down the rabbit hole, NADH and FADH2 do a lot. But for us, all we care about is that it contributes to the formation of ATP in the cellular respiration, which we'll be talking about in a little while. ATP again, wonderfulness. Now, how does ATP store up energy? Well, it does it via phosphorylation. Phosphorylation is when you're adding a phosphate onto it. We have, for instance, down here in this little graph right here, you'll see adenosine with two phosphates on it. It's ADP, adenosine di meaning two, phosphate meaning a phosphate. If you release a bunch of energy, let's say from sugar or fat or protein, if you have adenosine diphosphate nearby and you have the right enzymes going on for you, you're going to form ATP, adenosine triphosphate, which can now carry that energy to other places in the cell, which are really needing it. Maybe you want some protein metabolism, maybe you want some anabolism going on. But by breaking that bond between this dephosphorylating, is the appropriate term, dephosphorylating, breaking that bond will release that energy that you formed when making it. So anabolism, again, is to build up, and it does require that breaking dephosphorylation. So creating requires energy. Sweet little Aunt Anna needs her cup of joe. She needs her cookies and crumpets and all that wonderful deliciousness. A mean old cat releases energy. All that work can be broken down just to release that energy. Destroying liberates energy. So if you want to rephosphorylate that ADP into ATP, well, you got to get some energy from somewhere, and well, breaking glucose down into pyruvate's one of those things. So, quick This activity I'm going to go ahead and let you guys do on your own or in lecture. It's just a little way to help you, if you have the slides open, group these together based on whether they're anabolic or catabolic. Moving on. So the best starting point into our discussion in metabolism is carbohydrates. Carbohydrates meaning the complex sugars. Now why? Because pretty much everything feeds into it. You can feed it protein, you can feed fats into it, you can feed nucleic acids into it, and you can get energy from it and build things from it too. Now, in order for any of this to occur, this metabolism to occur, you do need to get that glucose into the body at first. And in order to do that, the majority of the cells need you to have insulin. Insulin increases the GLUT4, which allows GLUT4 to transport that sugar across the way. If you have type 1 diabetes, it means that you are lacking insulin, most likely because your immune system has destroyed the beta islet cells that create insulin. If you have type 2 diabetes, it's something wrong with the GLUT4, meaning that you've eaten way, way, way too much sugar. Maybe you just have some genetics not being your friend. Those GLUT4s become insensitive and no longer want to be present, and well, then your blood sugars start to increase even though you're trying to get sugars into that cell. Now that we're inside the cell, we've gotten through GLUT4, insulin's helped us out a little bit. We're in the liver for now, or maybe we're even in the muscles for now. If we were to wanting to make or break things up, there is something that we will do. We can make glucose into glycogen. What that is, so here's a picture of glycogen. You got right here in the middle of that glycogen, you got a nice little protein structure and all these little itty bitty dots with some blacks and reds and whites. Well, one of those itty bitty things is a glucose. So glycogen is a way for animals to store sugar, to store glucose. You're going to only store it in your liver and your muscles. Liver, you can pour it out, back out into the blood. Muscles, not so much. Some suffixes. Now let's go ahead and play. These terms that we're going to be talking about are important. They are going to be playing a role. You would do in tests and quizzes, so you better know all these. And thankfully, we've already talked about anabolism, catabolism, their suffixes, so this should make this a little bit easier. So when you break down glycogen, we know that lysis has to appear in the name, right? So there it is, glycogen. It's breaking down glycogenolysis. It's breaking down glycogen. If we want to make glycogen, we're going to, from glucose, we're going to put glucose on it. We go glycogenesis. It would be really funny to hear glycogenesis, but we don't really want the redundancy, so we combine the two gens together to just say glycogenesis. Genesis, remember, is to create. If we were to want to break down glucose, the term we would use is glycolysis. Not super clear as to why it's glycolysis rather than glucolysis, but please don't let those get confused in your brain. It is glycolysis. If we were to try and make pyruvate back into glucose, it would be gluconeogenesis. Remember, neo means new. So this is saying to create a new glucose, or to create a new glucose. I just said that. Oh well. Welcome to the video, a mistake. Now that we've talked about that, we can go ahead and talk about anaerobic, aka without oxygen. If your body, or primarily your muscles, decides that there's just not getting enough oxygen, but you still need that energy, what your body will do is it will take that pyruvate and it will modify it into something called lactic acid. It pretty much sounds like it is. It's not from milk, as some people think. It's just a different breakdown byproduct. That lactic acid, obviously, it's the thing that is somewhat responsible for that burning sensation in your muscles after a good long workout, and also the buildup of that lactic acid can persist for quite a while afterwards, or that pain from it can persist for quite a while afterwards. Your liver, though, can take that lactic acid and make it glucose again if need be. And again, that terminology from going lactic acid into glucose just lumps down into the gluconeogenesis term. Now, where they occur, kind of talked about it a little bit, but let's go ahead and talk about every single term. Glycolysis happens everywhere. It is the source of all energy. You will find every single enzyme responsible for breaking down glucose anywhere and everywhere. It is such a good way to get energy. For glycogenesis, you're only going to find it in the liver and muscles. Those are the only two places that you're going to store up sugars. Liver transports, lets it go for the rest of the body, but muscles holds on to it because, well, it needs that energy to be able to keep on moving even and not rely on transportation alone. Glycogenesis only occurs in the liver muscles, it better makes sense that breaking down glycogen only happens in the liver muscles as well. Fermentation, you're only going to find that mostly in your skeletal muscles. Fermentation is, again, reminder, when you are lacking oxygen. So it's got to be in the muscles mostly. Gluconeogenesis can occur in several places. It can happen in the liver, kidney, and astrocyte. Those of you that remember from AMP2 when we discussed the supporting cells, especially the blood barrier cells, you would remember that this astrocyte is that one that produces that blood-brain barrier. Your brain can only survive on glucose and run on glucose. So if there's lactic acid bouncing around or anything that can be made back into glucose bouncing around, you can bet your bottom dollar that that astrocyte's going to convert it into glucose because it wants to maintain that brain well, fed, and happy. Now let's switch over from anaerobic to aerobic, aka meaning you have oxygen floating around. You're going to be able to convert glycogen to glucose to pyruvate, but the main difference between aerobic and anaerobic is that you're going to have a cellular respiration in aerobic, meaning that you're going to break down these byproducts down into CO2, H2O, and ATP, and by golly, a lot more ATP. In fact, fermentation, you're only going to get 6 ATP out of it per glucose molecule, while in the cellular respiration, you're going to get 30 to 32 per glucose molecule, which is a major difference. That's 5 times more than fermentation process. This little equation you do need to know. So what happens? This is a redox reaction. It requires the destruction via oxidation of glucose into carbon dioxide, water, and ATP. You do need to know the numbers of each. Glucose. There's only one glucose. You need 6 oxygens and 30 to 32 ADPs, and 30 to 32 phosphates to make ATPs. That kind of makes sense, right? It's a 1-to-1 ratio there. What is interesting, though, that will hopefully make things quick and easy, is that if you have 6 oxygens, and the glucose, you're going to create 6 carbon dioxides and 6 waters. Another way to remember this is that glucose only has 6 carbons. So if y'all have taken chemistry yet, hopefully you have by this point, you have to maintain a balanced equation. So if you have 6 carbons here, you have to have 6 carbons over here. Since there's only 6 carbons here, that means you have to make carbon dioxide into 6. Let's break this apart now. We're going to go over the overview. We got 4 parts to our little aerobic respiration or cellular respiration. We got glycolysis, which we talked in great detail about the different processes a little bit. We're going to go dive into it even deeper. We got the formation of acetyl-CoA, so you're taking that pyruvic acid, that's the end of the glycolysis, to make into bind it to acetyl-CoA, and then feed it into a Krebs cycle. The Krebs cycle produces NADH, FADH2, and a bunch of hydrogens that eventually get fed into the electron transport chain, which is the primary maker of that ATP. The locations of which you do need to know. Notice that glycolysis occurs out in the cytosol. The conversion of acetyl and everything else that happens in this process occurs within the mitochondria. And yes, you do need to know every single part and where inside the mitochondria. I'll point it out again later on to help drive it through, but the Krebs cycle and the conversion of pyruvic acid into the conversion of acetyl-CoA does all occur within the mitochondrial matrix, with electron transport chain at the very last step happening using the intercellular membrane of this mitochondria. So glycolysis, here is the entire process of glycolysis. You're going to see that every single step requires some type of enzyme. It doesn't really mention a lot of these right here, but once you get down to when it splits that sugar in half, you're going to see that it has those enzyme, the names of these molecules, but in between every single step is an enzyme. So you have specific enzymes that only work with the specific bits and pieces by adding on or taking away. But what really is the take-home message? Well, for starters, there's a net sum of energy. You only get so much out of glycolysis. In order to make this occur, you do have to dedicate 2 ATP molecules, but in return, you get an additional 4 ATP molecules. You get a net gain of 2 ATPs out of glycolysis for every glucose molecule. Another thing that we can take or derive from glycolysis in this step, I do want to point out really quick that you don't need to know the names of all these enzymes and all these little molecules. We don't have time for you to do that, nor is it important. But as an FYI here, this is more for your pharmacology / help out with this. Again, these are commitment steps. These are commitment steps. You have put in energy. You have put your money into this process. You have paid to go into Disneyland. By golly, you're going to go ahead and enjoy it, whether or not you don't like the long lines and stuff like that. You're going to commit to it. You have put that energy. You have put that money forward. In order to reverse that requires energy. So not a happy place to be. Another one to note about drug metabolism and metabolism in general. Every single pathway has a rate limiting step, meaning what enzyme is the slowest? What is the slowest worker? In this case, it is the phosphofructokinase enzyme. The phosphofructokinase is the slowest moving enzyme. To give you an example of how important it is to know what is the weakest link and how to overcome it, this is kudzu. Kudzu has a modified enzyme. So plants can only grow so fast because of a rate limiting step called rubisco. That is a type of enzyme that has been thankfully shortened down for us because it's actually a rather large name. But because rubisco is really, really slow, and most of the time, plants, trees, grasses, name it, grow really slow. Kudzu, however, has a modified rubisco, which allows it to grow so super fast, ridiculously fast. It can grow a foot or 2 per day if left in ideal conditions. Back in Asia, where it originated, there's a lot of predators that eat it, so it doesn't have out of control things like it does here in North America, which was accidentally introduced by somebody thinking it was pretty and wanted to put it on their house. But now it's overwhelming and making things very, very bad for a lot of people and a lot of environment. Let's do a meta application. So drug metabolism. So there was a study in which researchers wanted to determine the metabolic rates of elephants and understanding it. And for some odd reason, they thought, hey, let's check cocaine. So they gave an elephant amount of cocaine. The cocaine has been well studied in humans. We understand their metabolic rates. We know how much can kill somebody. They know that you can't exceed a certain amount of cocaine per body weight. So you're supposed to give those of you that are in or are LPNs should already hopefully know that you're only able to give so much drug per pound. In elephants, they had determined that the human dose per pound in elephants should apply to them. Problem is, though, as soon as they administered it, the elephant died immediately. Why is that? Well, one, the elephant didn't really have the presence of those enzymes really well built up. Two, that enzyme inside the elephant that processes cocaine, well, it's a little bit slower. A lot slower, in fact, so it couldn't clear it out as quick. Which, by the way, if we were to take this and apply back to humans, that explains sort of why some people can not be phased or buzzed by ethanol, since they've built up tons and tons of enzymes in their liver to deal with the alcohol from probably constant exposures, maybe a little bit of genetics, while those that are brand spanking new, taking in some alcohol, can have a really tough time, get buzzed really quick type of thing. Krebs cycle. Look at all that pretty wonderness. We got through the glycolysis, there's pyruvic acid. We're going to do some modifications where we're going to take away one of these carbons and these carbon dioxide, one carbon, two oxygens, and stick it onto acetyl-CoA. Acetyl-CoA is going to feed it into the Krebs cycle. Again, you don't need to know oxaloacetic acid. You don't need to know the names. You don't need to know the names of any of this. All you need to know is, one, here's the location, again, within the mitochondrial matrix, deep inside the intercellular membrane, that also you're yielding out as part of your acetyl coenzyme transformation / Krebs cycle. You're making you're making all of your carbon dioxides. There's the 6 carbon dioxides. You're making a net amount of 2 ATP here, as well as you're making NADH and FADH2, which, if you remember from the beginning, contributes to the electron transport chain to help create the most ATP you can possibly make. Important notes about the Krebs cycle is protein metabolism. This is where you can feed in a lot of the proteins, amino acids, the base building blocks of proteins. So proteins can be broken down, destroyed, or even built up in a way from these building blocks. Here's a quick FYI, by the way, the byproduct of amino acid breakdown into this cycle is urea. Hopefully that sounds familiar. Urea is found in your urine. It gives your urine that yellowish color. Now, also another FYI, which will be important for you later on in nutrition and all that stuff. Y'all heard about essential versus non-essential amino acids. All amino acids obviously will be degraded. However, there are ones that cannot be made. You can't reverse the thing and make it anew. You can't make some amino acids anew. You have to eat it. You have to get it from your outside environment, from things that can make it. Non-essential? Yeah, you can make it. You can reverse the process and you get it. Sure can. Why not? This is one of the hurdles behind when people started becoming vegetarian and understanding the principles of trying to be vegan. And they didn't understand that there were essential amino acids that they needed to eat. And some vegetables didn't have those essential amino acids in them, which eventually caused some very serious issues in their body as their body slowly cleared out all their essential amino acids. Now, if we combine both Krebs and glycolysis, we can talk about lipid metabolism. You'll see that here, that again, there's the amino acids, but you can see that glycerol and fatty acids and those triglycerides can be converted and moved around on the outside, but can be brought into glycolysis to be modified and put into the cycle to be made into ATP You could also put in those fatty acids to be inserted into the acetyl coenzyme factor, but you will also notice that you can make ketone bodies out of this. Ketone bodies should sound somewhat familiar because, well, diabetic ketoacidosis derives from this, meaning that if you are having issues with insulin or if you are having issues with your GLUT4 receptor and you're not getting enough sugar into your body or into your cells, and they're screaming for energy, they are screaming for that glucose, but for some odd reason can't get it. What your liver will do is it will create a bunch of ketone bodies which sneak into the different cells in different ways, where you can sneak it in into the Krebs cycle, you can sneak it into the acetyl coenzyme and all that stuff to get that energy. Your brain only uses these ketone bodies when it's in starvation mode, by the way. This also explains why you can smell that fruity smell in someone's breath. I call it more of a toluene smell, acetone smell that you're going to smell in someone's breath when they're going through diabetic ketoacidosis. Finally, the last step in this video is the electron transport chain. Here's the very, very basic principles of it. NADH brings hydrogens to this inner mitochondrial membrane. This entire process of multiple enzymes, multiple protein channels, put hydrogen between the outer and inner membrane of this mitochondria. By doing so, they are making a proton concentration gradient. And these things want out, and they want out now because, remember, no one likes to be in heavily concentrated areas, especially with COVID. That's not going to apply in a year or two, but here we are. Hopefully, it won't apply a year or two from 2020, but we'll see. So this high concentration of hydrogen ions looks for a hole to escape, and it finds it through an ATP synthase, which when that hydrogen circulates around in it, causes it to churn, which develops a type of energy that is attached to an ADP, which allows for ADP and phosphate to phosphorylate to create that ATP, which creates the bulk majority of your ATP. It creates the most ATP. It creates 26 to 28 ATP molecules per glucose. That is amazing how much it can do. Where does it occur? There it is again. There's that ETC, aka the electron transport chain. You can see it. Occurs across the inner mitochondrial membrane here. Here it is in a little bit more detail, now that we know and understand it. So NADH is going to feed in a hydrogen and at the same time an electron, hence electron transport chain. That electron transport chain gets passed along multiple enzymes that just describes a few of these here. You don't need to know their names, but the electron transport chain sort of teases at that hydrogen. And that hydrogen follows along with it, along with its proton gradient. And then at this very end point, this electron gets booted out. Hydrogen comes chasing after it, along with its concentration gradient pushing at it. So it's a double whammy, which generates a bunch of energy, enough for an ADP plus the ADP to be converted into an ATP. In order to store that hydrogen so it's not acidic, as well as to find a place for that electron, the hydrogen ions bind onto oxygen, which accepts both. So the hydrogen and the oxygen both accept this electron and allow the formation of H2O. This is metabolic water. This is you making water. Obviously, this isn't enough for you to survive on your own, but it is metabolic water nonetheless. So this is our last slide. That'll summarize everything that we just talked about. Glycolysis over here. Glucose 6-phosphate. If you get glucose from the blood, it modifies it into glucose 6-phosphate. If you get it from glycogen, glycogen can be modified into glucose 6-phosphate, which can be inserted into glycolysis. Glycolysis makes pyruvic acid. If you want to make it if you do not have oxygen present, you go through some anaerobic reactions, fermentation primarily, which forms lactic acid. If you do have oxygen present, then it is moved into the Krebs cycle, where it forms and produces not only carbon dioxide, but NADH and FADH2, which can then be turned into the electron transport chain, which produces that high amounts of, or aka 26 to 28 ATP molecules per glucose molecule. Okay, I lied. There's a couple more. There are 3 more slides that we're going to talk about. Now that we know the process, we know about how everything works, how fats and all that stuff moves and is used, we can point out a couple of things. Glucose, again, is absorbed via the small intestine, along with amino acids, triglycerides. Triglycerides we're going to be talking about that in another slide, are stored up in fats until they are needed and used again. But the liver can convert all of the glucose, ketone bodies, fatty acids back into one another. And that is how we will end that. This gets a little bit more complicated. Again, fatty acids can be converted into the varying types of things, including glucose, which can be shot out into the body and used in different ways. Here are the different things that can use it, and the other things. The heart can use fatty acids, lactic acids, and ketones to make ATP. Other tissues, let's see here. Muscles, let's just go over to muscles. Muscles can donate amino acids, but it can take up that glucose. Again, remember that glucose is used everywhere and anywhere. With that, that is the end of our discussion. Again, here's the end process. 1 glucose molecule, 6 oxygen molecules with the necessary components for ATP to be made will yield out 6 CO2 molecules, because you have 6 carbons here, 6 water molecules, and 30 to 32 ATPs. That is probably the longest video I've ever created. Sorry, but here we are. Take care, y'all.